Assalamualaikum everyone, we are the students of Osh International Medical University and today we are presenting the Neoplasia. It's me, Mr. Ali Raza, student of Osh International Medical University, and today I have decided to tell you about the Neoplasia. Actually, what is Neoplasia? Neoplasia is the uncontrolled or unregulated cell growth or new tissue growth, that is called as Neoplasia. And also in Neoplasia we are having two types of tissue involved. The first one is the neoplastic and the second one is non-neoplastic. Now, for example, what is the neoplastic tissue? Neoplastic cells are called the parenchymal cells. For example, if you take the example of the liver, in liver the parenchymal cells are hepatocytes, right? And the next one is non-neoplastic cells. Basically, non-neoplastic cells means supporting cells, the supporting cells of our body. They are called non-neoplastic cells. And the next thing is the, for example, mutation. Now, what is mutation? Any change in tiny part of DNA is called as mutation. Okay. So the next one, for example, we'll talk about the types of the neoplasia. The first one is the benign, as you can see, and the second one is malignant, right? If we talk about the benign tumor basically they all end with suffix oma, but there is some exception. And for the exception, we have memorized a mnemonic LM2 NGHS. Okay, the very first one is, for example, lymphoma, mesothelioma, melanoma, glioma, hepatoma, neuroblastoma, and seminoma. You can see that these type of, for example, tumors, they are all ending with suffix oma, but still they are categorized as malignant tumor, right? Okay, so the next one is, let's talk about the malignant tumor now. Okay, basically malignant tumor is of two types. The first one is sarcoma and the second one is carcinoma. Now, this sarcoma, it is the tumor of the connective tissue or mesenchyme. And carcinoma, it is the tumor of the epithelial cells, especially transitional epithelial, squamous epithelial or glandular epithelial. If we talk about this sarcoma, as we already have said that it is the tumor of connective tissue, 40% of the sarcoma is present in the lower limb and it has high vascularity, right? And if we talk about the carcinoma, yeah, we are having the transitional squamous and glandular epithelium, right, for this. And if we talk about the features of the benign tumor and malignant tumor, basically in benign tumor, we are having the stroma or capsule. And this capsule will not let the benign tumor to spread in our body, right? And if we talk about the malignant, so in malignant we are having metastasis. Metastasis means, for example, the tumor cells, they spread in our body from one place to another place, right? Except the glioma, the rodent ulcer or basal cells. Except these three cancers, the rest of all the, for example, malignant tumor, they all metastasize, they all spread in our body, right? And the next feature of benign tumor is that it is organized, and the next feature of malignant tumor, this is disorganized. And in benign tumor we are having decreased mitotic activity, and in malignant tumor we have increased mitotic activity, and also mostly in malignant tumor we are having necrosis, right? And the next one, in benign tumor we do not have local invasion, means local invasion is absent. And in the malignant tumor we are having local invasion and also loss of polarity.
Bismillahirrahmanirrahim. My name is Muhammad Salman. I am student of in Osh International Medical University. So now we will discuss about the features of neoplasia. Some characteristics. First of all, monoclonality. Monoclonality, in normally what happen from one cell from parents, means one cell from mother and one cell from father goes to the cell division. But in monoclonality, single mother cell goes to cell division. And for example, we have example that glucose 6-phosphate decarboxylase. It has the three isotopes A, B, and C on the chromosomes X. Generally, if we talk about females, in female there are the two X chromosomes. If one chromosome X is inactive, this term is called lionization. And the second example is B lymphocytes. B lymphocytes, for example, we have B lymphocytes is made up of light and heavy chains. And heavy chains is made kappa and lambda, which ratio is normally 3:1. But when neoplasia is occur in monoclonality, example B lymphocyte, the abnormalities in ratio of B lymphocyte is 6:1 or 1:3 maybe. And next is anaplasia. Anaplasia, ana means not, and the absence of differentiation. We cannot differentiate between normal cell and mitotic cells. Maybe it normal cell or neoplasiatic cell. For example, there are three forms. That is, number one is pleomorphism. Pleomorphism, basically it is the change. It is a change in size or shape of cell. And the next one is hyperchromasia. Hyperchromasia, that is the dark staining color of DNA. And next one is nucleus to cytoplasmic ratio. Normally, nucleus to cytoplasm ratio is 1:4 or 1:6. But when neoplasia occurs, its ratio becomes to decrease. That abnormal ratio is 1:1. And the main features of nucleus to cytoplasmic ratio is it increase the mitotic activity and also it decrease the cell polarity. Next factor is that growth rate, which is inversely proportional to differentiation. For example, if differentiation is increased, growth rate is decreased, and growth rate will be decreases, differentiation increase. If differentiation increase, that means there are high differentiation in normal cell and abnormal cells, so the proliferation will be slow. If the differentiation decrease or undifferentiation, proliferation may be increased or high proliferation. Next one is local invasion. Local invasion may be divided into, it will be divided into 4 steps. Maybe number 1 steps we have that eke adherence. Eke adherence, that firstly the mitotic cell or neoplastic cell is attached to the normal cells. For that is eke adherence, means loosely attachment between the cells. Each cells are loosely attached each another. Step 2, we have attach or degradation of basement membrane. It is very important features clinically or medically. Clinically, the attachment or degradation of basement membrane through the enzyme that are hydrolase and collagenase. They are the enzymes that which helps to the attachment or degradation. So there is second step, the attack or degradation of basement membrane through the enzyme that are called hydrolase or the collagenase. And the medically or clinically it is important because there is a carcinoma that are in cytocarcinoma which does not degrade the degradation of basement membrane. They are the steps. Step 3, that is attachment or degradation of ECM, that is extracellular matrix or extracellular membrane. Then step 4, it increases the cell molality. There is a term that intravasation base. It means that when the cell motility is increased, means cell movement is increased, and it attached to the capillary. Then after capillary, it may be enter to the capillary, goes to the blood, and the term is called intravasation. So the last one, but not the least, that one is metastasis. Metastasis, basically it is the spread of tumor cells throughout the body from one organ to another organ. There are the 3 steps, there are the 3 conditions for the metastasis. Number 1 is lymph vessels. Lymph vessels may be lymphatic system, maybe through the limbs, maybe tumor spread from one organ to another organ. Second one is tumor cells spread through the blood vessels. And last one is blood cavity, body cavities. Body cavities include tumor cells spread through the body cavities, maybe cranial cavity, thoracic.
My name is Muhammad Numan. Today I will tell you about the screening. The goal of screening to catch the dysplasia. Dysplasia means disordered cellular growth. It is precancerous and irreversible neoplasia. Cervical defect, cervical interrupt neoplasia, CIN, will do pap smears. So It will do mammography for in situ breast carcinoma and ductal carcinoma in situ DCIS. So it will do Paget's carcinoma of nipple in which eczema of dermatitis. Prostate specific antigen or digital rectal examination will detect the prostate, and colonoscopy to detect the colon. Epidemiology. It is the second most common death of male and female and children are cancer. First most common cause of death in female and male is cardiovascular disease, and second most common cause of disease death is cancer, and third is chronic respiratory infection. It is first most common cause of death in children accident, and second most common cause of death in children is cancer, and third is congenital defect. Incidental rate. First in male in first prostate, then lungs and colorectal cancer, and second is female breast, lungs, and then colorectal cancer. Mortality in male, first is lungs, then prostate, and then colorectal cancer, and in female, first is lungs, and second is breast, and third is colorectal cancer. Thank you so much.
My name is Tamurli. Today I will talk about characteristics or feature of neoplasia. Telomeres activity. Telomeres is a basic part of DNA which protects chromosomes to damage, and each time when a cell divides, its length becomes shorter. It also plays an important role in the age or lifespan of human beings. The other part is upregulation of decay accelerating factor which inhibits the membrane attack complex. Due to the inhibition of membrane attack complex, there is no lysis of cell, so there will be a more and more regulation of decay accelerating factor which inhibits the membrane attack complex. Due to the inhibition of membrane attack complex, there will be no lysis of cells, and there will be more mitotic activity of cells, and there are more and more new cells will be generated. And the other point is cell organelles. It includes Barber effect which shifts of mitochondrial oxidative phosphorylation. Another topic is cell organelles. Decreased mitotic activity or decreased of endoplasmic, which is Barber effect, shift of mitochondrial oxidative phosphorylation to anaerobic glycolysis. It also a feature of neoplasia, and other is anaerobic glycolysis which increase the production of lactic acid. And there are other tumor-like conditions in which hematoma and tumor-like condition. First is hematoma and second is cholestoma. Hematoma is abnormal tissue growth at normal sites like bronchial cartilages and glandular tissue, and other is cholestoma, which is the normal tissue growth at abnormal sites like pancreatic tissue and stomach and brain tissue and nasal cavity. Thank you.